Welcome everyone. Here is another question from GATE PYQ series. The question is, a cytoplasmic male sterile female plant with the restorer genotype small r is crossed to a male fertile male plant with the genotype capital R capital R. Both capital R and capital R small r can restore the fertility, whereas small r cannot. When an F1 female plant with the capital R small r genotype was test crossed to a male fertile male plant with the small r genotype, the percentage of the population that is male fertile would be, we need to calculate the value in percent. So before solving this question, let's try to understand what is asked. So this question deals with cytoplasmic male sterility, and this is a phenomenon where the plant cell is unable to produce the functional pollen. So the male is sterile, and the mode of inheritance is cytoplasmic, as it is clear from the name. And this is maternally inherited, that is often associated with the unusual open reading frames found in the mitochondrial genome. So dysfunctional mitochondrial genome results in the male sterility in the plants, where the plant cell is unable to produce the functional pollen. Also, there are certain information which we need to understand. So if a plant is carrying dysfunctional mitochondrial genome, that will result in the cytoplasmic male sterility, but there is another gene which comes from the nucleus, which can restore the fertility, and this is called as the fertility restorer gene, RFG. So the presence of a single capital R is sufficient enough to restore the fertility, even if their mitochondrial genome is dysfunctional, resulting in the CMS. So summarizing the things, sterility results from mitochondrial genes causing cytoplasmic dysfunction, while on the other hand, fertility restoration relies on nuclear gene that suppress the cytoplasmic dysfunction. So this is the nuclear gene, and this is the cytoplasm. And this CMS gene is coming from the extranuclear gene, and we all know the source of the extranuclear gene in the plants are mainly the mitochondria and chloroplasts. And we all know that progeny inherit cytoplasm from the mother. Keeping all this information in mind, let's solve this question. So what is given? A cytoplasmic male sterile female plant. So this is a female plant, and the cytoplasm is CMS with the restorer genotype small r small r. So the one which is coming from the nucleus is small r small r. Remember, this is not sufficient enough to restore the fertility. So it is crossed to a male fertile male plant. So this is male plant. So this is fertile, so no CMS. And the genotype of the nuclear gene is capital R, as I just now said, a single copy of a capital R is sufficient enough to restore the fertility. That means in that case, the plant is going to be fertile. So when you are going to make this cross, the progeny is going to carry, remember, first we'll consider the nuclear gene. This parent will only give capital R, and this parent will give small r. And we all know the cytoplasm comes from the female parent. So the cytoplasm in this case is going to be the CMS, but the nuclear gene is going to be capital R, small r, and this progeny is going to be fertile. The reason we all know, because the presence of the capital R is sufficient enough to restore the fertility. Now, what is the next part? When an F1 female plant with the RR genotype, so you have this female plant, nuclear gene is capital R, small r, and this is the cytoplasm is going to carry the CMS gene, was test cross. Test cross is a kind of a cross where we make use of a homozygous recessive parent. And also it's given to a male fertile male plant with the small r genotype. Small r genotype. And we are not bothered about the cytoplasms from the male parent because we all know the female plant is going to pass the cytoplasm to their progeny. This is female, this is male. Anyways, it's given the male is fertile. Now, what is asked is the percentage of the population that is male fertile. Remember the kind of the gametes at the level of the nuclear gene. This parent is going to produce is capital R, small r, and this parent is going to produce a small r only, 100%. So the progeny is going to be, so they will inherit the cytoplasm from the mother. So this is going to be the CM. But the most important part is what is the genotype going to be for the nuclear encoded gene. So half of the progeny are going to carry capital R, small r, and half of the progeny are going to carry a small r, small r. As I said, they can only give either capital R, which can combine with the small r, or in the other case, the small r can combine with the small r. So 50% this kind of a genotype and 50% of this kind of a genotype. Now remember, presence of a capital R is going to make this progeny fertile. Even though the cytoplasm carries the dysfunctional gene, mitochondrial genome, which is resulting in the CMS, but the presence of the capital R in the nucleus results in the restoration of the fertility. However, in this case, this plant is going to be sterile because there is no presence of a single capital R in the nuclear genome. So the final answer is going to be the percentage of the population that is male fertile would be 50%. I hope this answer is clear to each and every one of you. If you do have any doubt, please feel free to comment. Thanks for watching the video.